Julianne, and welcome back to the sixth and final talk in our summer series of the broad-reaching work of the Florida Museum of Natural History. Last week, Dr. Ed Stanley told us about using computerized tomography to investigate the skeletal structure of small animals, some even the size of a flea. I think those of you who are here remember vividly that demonstration. This week, Dr. David Blackburn is going to talk about using similar 3D imaging techniques using visible light to make the content of the jars that are hidden in the back room of museum collections accessible to scientists and classrooms around the world. Dr. Blackburn is the curator of amphibians and reptiles at the Florida Museum. He studies the diversity and evolution of amphibians, especially frogs. His field of research is focused on Central African tropical forests for the past 2 decades, including descriptions of many new species. And he spent the last 5 years leading a collective project involving 25 US institutions that are working together to create high-resolution 3D digital anatomical data freely available for online for all living vertebrates. A challenging job. The title of his talk is A Cloak and Dagger Story, a riveting, get this, riveting tale of making hidden museum collections more accessible. So please welcome Dr. David Blackburn.
Great. All right. Thank you very much. It's really nice to be here. We'll wait for our slide to show up. So we had a challenge presented from last week when Ed Stanley, who is a longtime friend and collaborator, came to talk, because we had to partition our talks because I was not expecting him to give a talk. So we will talk about some CT scanning. We'll see some of the same types of things, but hopefully I can go more in depth to talk about especially some of the non-research uses of these data. So I came to the Florida Museum of Natural History 7 years ago. Before that, I was at the California Academy of Sciences, which is the Natural History Museum in Golden Gate Park. Most of my work, as John mentioned, is on frogs. So I'm going to talk just briefly about some of the frog science that I do and sort of what drives that, and then use that to explain a little bit about how I got involved in these other projects that don't have anything to do with frogs, and give you a sense of really where that work is taking us in the museum. See if I can do this correctly. And I was challenged to see how many puns I could put in a title. That was my best, so I have 2. Okay, so I work on frogs. There's currently about 7,500 or more species of frogs. We describe new species of frogs as a global community of scientists. We describe several species a week, right? So this is an exciting time, which I'll come back to and say more about. Not only are these lists, they're species on a list, but of course, when we're discovering new species, we're discovering new biology, right? We're discovering new shapes, sizes, colors, life histories, and so that sort of richness of frog biology is what attracted me to do what I do, and to work on amphibian diversity. So one of the things that's interesting about being a frog biologist, I'm regularly told by people that they're terrified of frogs. So some people are terrified of frogs. Frogs also have had a pretty bad rap in Western civilization for the last 2,000 years. The Bible only has frogs appear twice, in the beginning and the end, basically. First as a form of biological warfare, and second, coming out of the false prophet's mouth. I would say neither of these are very compelling if you want to attract people to work on frogs, right? And it has been a difficult road for many frog biologists over the past few hundred years. But interestingly, frog science was rehabilitated in a way by this Dutchman, Jan Swammerdam, who really in the 17th century started using microscopes to look at the insides of animals. He's most famous for working on insects. However, he also worked on tadpoles. And part of the interest there was at the time, amphibians were thought to maybe not really be sort of animals like us. They were thought to sort of appear out of the muck, spontaneously generate. It was not thought that, tadpoles and frogs were even what we now think of as a vertebrate animal. They were thought to be quite different. But Swammerdam, let's see if I get this right, showed in these dissections of tadpoles that, in fact, they had many of the same parts that we do. They have a heart, they have lungs, they have stomach. So one of the most exciting and interesting things from Swammerdam was that, you see these plates in this classic work that was published long after he died. These little diagrams are actually pieces of frog muscle dissected out of a frog, and then they were doing experiments with it, right? These were early experiments to see, how did muscles work, right? But it was all predicated on the fact that, well, frogs had something in common with us, and so taking a piece of them out and studying in it somehow informed, what we think about our own bodies and how our own bodies worked. I think this is pretty amazing, actually, that this is happening at this point. This is printed, decades after he died. So this is really happening in the 17th century. But if you fast forward to now, frogs have been really important in our understanding of general biology. So, as an example, the very first cloned vertebrate was a northern leopard frog. The very first vertebrate born in space and then returned, so you might consider them the first aliens, were frogs. They were African clawed frogs, like this guy. Oh, wrong button. This guy here. And there's been more than a dozen Nobel Prizes that have somehow involved frogs as part of their science. And so most classically in the last few decades, despite the sort of early work on leopard frogs, like this guy here, we have a species of leopard frog in Florida. A lot of the biology has been on this aquatic frog from Africa, the clawed frog. These are used in biomedical research all over the world. They've basically, beginning in the 1930s, were used very widely, in part because there are two reasons that frogs started getting used in research that today resulted in Nobel Prizes. So one was that when they lay eggs and they have fertilized eggs, they don't have a shell. So you could actually see all of what's happening in development just from looking through the egg membrane. So it was easy to observe. But the other thing that was discovered was that you could expose these frogs to a hormone and make them lay eggs on command. So for that reason, just out of curiosity, does anybody have any memory of going to a doctor's office as a kid and having frogs like these? One of my older colleagues who passed away last year said, he grew up in South Dakota, said that he has a memory of the doctor's office having these frogs, and because they were used as pregnancy tests. So if you expose them to a pregnant woman's urine, the hormone that's in the urine that's used for pregnancy tests, which is chorionic gonadotropin, makes these frogs lay eggs, because it's basically the same hormones in their body as in our body. And so because you could manipulate their egg laying so easily, they were used not only in pregnancy tests for a little while, but they were also used widely around the world for developmental biology, because you could have them lay eggs on command, right? Which is very useful if you're interested in studying that. Today, that tradition has continued in different ways. So these are wood frogs. Wood frogs have this amazing ability to freeze solid, have their heart stop, thaw, and resume, which is remarkable. So these are, one of many other frog species that today are actually used in biomedical research for, we're learning interesting things about the biology of these animals, but we're also hoping to apply that sort of knowledge to ourselves in some way. So as I noted, this is an exciting time to be an amphibian biologist. So just to date myself for all of you. So I was born in '79. There's well more than twice as many amphibian species today as there were when I was born, which I think is totally wild when you think about it. So we describe as a community 3 to 5 new species of amphibians every week. That's mostly frogs. So there's 3 types of amphibians. There's frogs and toads, that counts as one, salamanders and newts, and then there's this third really poorly known group. There's only about 200 species. They're called caecilians. I used to say we don't have them in the United States, but now we actually have a species that lives in the canals of Miami that was introduced there. So we even have those in Florida now. So this is a pretty remarkable time, because not only are these new species that we're just adding to a global inventory, but we're of course learning all sorts of new ways about being an amphibian through these discoveries of new species. And wait. Oh, I'm sorry. Okay, good. Sorry. Please interrupt me. All right, that's fine. Okay, so one of the reasons that I became interested in studying frogs is, as a biologist that studies evolution, I've been interested in convergent evolution of very different groups of animals evolving to look the same. And an exciting thing about frogs is this seems to happen all the time. They basically take a common body form and evolve similar things again and again and again and again. So this is just a sort of a classic diagram that appeared in a paper in the late 80s. And it sort of showed in a cartoonish way these sort of differences between, if you have really long arms and really long legs, they tend to be these jumping frogs, tend to be tree frogs. Things that tend to have really short legs and arms are these burrowers, and then there's everywhere in between. And what's kind of neat is if you look across frogs, you find plenty of examples of this. But what's kind of hidden between just the appearances here is that each one of these pairs that I put up here, they shared a common ancestor more than 150 million years ago, which is stunning when you think about it, right? And so not only have frogs been evolving, tree frogs many, many times, but they've also done so over an extremely long evolutionary timescale. So I came from a background in paleontology, and this was part of what attracted me into thinking about frogs as a group of animals that I wanted to study. One of the things that's kind of nice, in a way, is that frogs have a very simple skeleton. Pretty much all frogs share a lot of the same features. And no matter, just as one example, no matter what frog you look at, the bones in their forearm, which are here in us, and the bones in their hind leg, which are the shin bones for us, those are fused together in every single frog. It means that they're very instantly recognizable in the fossil record for 200 million years because you can see those features. So one of the things that I'm interested in my lab is understanding how you take this sort of common, simple body form, and then you evolve all of these different forms repeatedly again and again. And the way that we do that in my lab, not always, but tends to focus on the skeleton. And so these are just examples. These are skulls of frogs. These are upper arm bones of frogs. These are bones of frogs in comparison to fossils. And so bones in my lab tends to be the currency that we use for making a lot of discoveries related to the fossil record, to how traits evolve, to how organisms work. So we also do a lot of functional biology to understand how animals are moving. But throughout all of those projects, bones is the thing that's important to me. So I'm going to segue here. So the question is, how do we get these bones, right? So when I started graduate school, we had a skeleton collection. I could actually compare, some of the skeletons that I was studying to some of the other skeletons that were in the collection. This was a very good skeleton collection. So we had maybe 10,000 dry skeletons from around the world. But we still didn't have lots of families of frogs represented. We didn't have all of these new species that were being discovered represented in that collection. So it made it very hard to make thorough comparisons between what I had in hand to all of these things that were found around the world. So the way I got into this business of CT scanning a lot of things was trying to actually build up a library for us of all of these skeletons representing frogs from all around the world. And thus, I'm now going to talk about the rest of what my lab has been spending time doing. So throughout a lot of my career, I've spent time doing field research in tropical Africa. While it sounds exciting and sexy, you might think, well, you spend a lot of time hiking around at night, which is true. We do go out every night, usually until our bags are full of frogs. We actually spend the majority of the time like this, sitting around actually processing samples, which can literally take us 10 hours a day. We might go out for 2 hours a night and then work 10 hours the next day, basically just working through that. And that's because for the specimens we create that we bring back to the museum, we're taking photographs of them, like student Brian is doing back here. We're taking samples of their skins or insides for screening for diseases. We're taking tissue samples for doing genetics and genomic science. We're also preserving these animals so we can later do anatomical studies. We take a lot of information from every one of these animals that comes back to our collection as a preserved specimen. And these end up resulting in these things. So here is a specimen. It's a scientific specimen. It has a field tag on it for my friend and colleague in San Francisco, Jens Werner Windum. It has a number on the other side. This is a frog specimen from our field research in Angola that we've had a project going in Angola for many years. And these are the currency of much of the research that we do in the lab. We use specimens, and in the museum at the Florida Museum of Natural History, we have 40 million objects or specimens. Those might be, ethnographic objects, things that are, we have a Seminole doll collection, for instance. We also have 200,000 preserved amphibians and reptiles as well. So we have a lot of these in our collection. Now, to be a scientist and to access these things, we normally go here. This is what our library looks like in the museum. These are just a library of jars, things in jars that are organized mostly by species. These are, in amphibians and reptiles, we preserve things with formaldehyde and then we store them in alcohol, much as has been done for most of the last 200 years. So we have a library of things in jars. And if we're a scientist that wants to study them, we can, visit the shelves, open the jars, work on these animals. They're used for dissection to understand what animals ate. If I want to examine specimens that are at another museum on the other side of the world, I have two options. I can either visit the museum or I can have them send it to me. Now, there's reasons that both of those are impossible. So one of which is that for many parts of the world, we will probably never put the specimen in the mail to them. I've worked for 20 years in sub-Saharan Africa. There's maybe 2 countries that we would actually mail specimens to and think that they wouldn't get lost along the way. South Africa is the main one that we would send material to. So if you're, and any of my colleagues I've worked with for decades now in sub-Saharan Africa, we're probably never going to send these specimens in the mail to you, and just because they will never arrive, which is a problem. The other, of course, is that it can be very costly to get on a plane, fly around the world to look at 8 dead frogs, which seems like a lot of money sometimes. So, these collections are here. They're here to be used, but they're costly and expensive sometimes to access. So I'm going to take one step back. So it used to be, so this is one of what's often acclaimed to be one of the very first natural history collections. This is the Wunderkammer of Ulysses Aldrovandi. Pieces of this still exist. I actually was able to visit this in June on vacation in Italy. So the University of Bologna has some parts of this collection are still there. But, in practice, this was the whole collection. You could walk into the room and you could see everything at once, which is sort of remarkable because today, our collections look like this, This is just a snapshot, a very famous picture of bird collections from the Smithsonian Institution. As a curator, I probably stood a chance of laying hands and eyes on every specimen in the collection if I was Aldrovandi, but it's impossible today, right? And so the way that we know what we have, instead of being able to see it at a glance, is that we have created digital records of a lot of this information. This is sort of a mind-numbing map, but just to orient you, this is a map of the world, and it's mapping just the points on a map of all of the museum specimens from around the world that we could assign a latitude and longitude to, and it's sort of a heat map of showing, where we have the most records. So you see some of these bright red points. There's more than 500,000 specimens, plants, insects, frogs, that are in those points. Right, so there's more than 130 million of these records you can quickly find on the internet, in a matter of moments, really. However, most of the specimens are just as inaccessible as they were to Darwin. right? And the reason is because most of that information I showed on the map is just a description. It's what's the name of the species, where was it found, who found it, when did they find it. It's not a representation of the thing itself. There's often not images. Typically for vertebrates, there's almost never images. For plants, it's easier to take pictures because they're two-dimensional on a sheet of paper. But the access to these things is just about as bad as it was to early naturalists. So what we have spent a lot of time doing in the lab, and Ed, who you saw last week, played a very important role in, is creating these sort of digital specimens. With digital specimens, we can email the specimens around the world quickly, which is something that we do pretty regularly. Just as a quick aside, India, for instance, is a good example. So India, for many years, has been very difficult to export specimens, even from museum to museum. You might think, well, I'm in a museum. There's a museum in Calcutta. How hard could it be to ship the specimens between two museums? Very hard is the answer. Sometimes impossible, both because of just personnel and the mail, but also because of international regulations. I wanted every frog that I could get my hands on for our project. So we actually sent Ed to Pune, India, where we knew there was a CT machine. And Ed spent all night CT scanning frogs in India with our collaborators, and then just literally was emailing me images while I was sitting in my office. And so, we can create these digital objects that get the job done for a lot of what we want to do. And while I'm going to talk some about research, the thing that's also really important, as you probably know from the museum, is that education and outreach, reaching a general larger audience, not just scientists, is also a really important part of museums. And so part of what attracted me to making these sort of digital specimens is being able to expand, who is it that interacts with these things that we care about. We spend a lot of money caring for these specimens for not only, decades, but even generations. We want these to be accessible to as many people as possible. So I'm going to come back to this frog. Ed might have shown you this. That's fine. This is Ed's image, but it's a great one. So here's our normal image of a frog. We can use CT scanning, which is basically just fancy x-rays, for creating all of these different representations from this animal. We can see its skeleton, its skull, insides of it. This is a representation of its inner ear. This is just a space. It's not actually a thing you could hold. It's just the absence of a thing, right? This is the space inside the inner ear. We can create digital models that represent, how thick the skull is, how dense it is, things like that. We can also 3D print models from this, which are used in education and classrooms. But you might be interested in other parts of the animal. So if we soak these preserved specimens in iodine, it makes their soft tissue dense to X-ray. If any of you have ever drank sweet syrup at the doctor's office to look at your intestines, usually has barium inside it. So the barium coats the inside of your gastrointestinal tract, and thus you can see it in an X-ray because the inside is now dense to X-rays. We can't make these dead frogs drink barium, so instead what we do is we soak them in iodine. And the iodine enables us to see skins and muscles and nerves and the heart and the cardiovascular system, even eggs and parasites inside of them. So we can do an awful lot with just a single specimen we've spent time creating these data for. So to give you just a quick sort of overview, when I say 3D data, we actually mean three different types of things. We mean these sets of two-dimensional X-rays that look just like this, right? These are just 2D X-rays. From those, we can actually create 3D models. The 2D X-rays are interesting and useful. Obviously, you can see the skeleton here. But once we create 3D models, we can start visualizing and manipulating a 3D model, even just online, in a web browser like Chrome or Safari. We create these, sorry, wrong button. We create these models like this. These are effectively just maps of points. They're just, all it is a computer file that shows how all of these points in three-dimensional space are connected to one another. That's all the computer file really is. And we can also create these other files like this that basically are a picture that we can wrap around that 3D model. And so these three things in combination are our data. And we can do a lot of things like both for science and also for education and outreach. So just to show you how do we make these different types of data. So one of them is a CT scanner. So at the University of Florida's nanoscale research facility, which is part of the College of Engineering, we have actually two CT scanners now. One is for extremely high resolution. That's actually, I think Ed showed you lice or fleas last week. That's really his workhorse for these extremely tiny things. For frogs, we don't need to do that quite as much. So we use this machine. We create these 2D x-rays, and then from that we can make these 3D models. But it's also very expensive. So for cost, this machine costs us about $800,000 to $900,000 to buy in the end. And that's not counting the maintenance costs and all those other costs associated. We have two other ways that we create these data in the lab. So one of which is that we can take what looks like a cheap plastic iron, it shoots out light from it, and it bounces back to a little sensor. And from that, we can actually create 3D models of things like, here's a porpoise skull. Those are great for big things, for hard things that are not shiny and hairy. We can create 3D models very quickly and easily of fossils, of big skulls of mammals, things like that. Things that we could actually never fit in a CT scanner. So, we can fit something about this big in a CT scanner, but once it gets bigger, we can't do that type of thing with this technology. So we use this machine a lot. We have a lot of undergraduates like Victoria who are trained up and actually generate all of these data for us, are mostly students in the lab. And the last, this is a little harder to see, but here's a camera, here's a camera, here's a camera, and here's a skull. So we can actually just use several, quote, normal, nicer cameras to take images all at the same time of an object, and we can use those to create these models called photogrammetry models. The value here is that you could technically do this with just one camera. It's just faster if we use several cameras. So this is relatively cheap, but it only shows the outside of the animal. This is quite expensive, but we get all of these other data as well. And so actually we use all three of these things now together in order to sort of make these digital representations of the things that are at the museum. I'm showing you, because I'm very biased, frogs and lizards, but of course we're also doing this with pottery. We're also doing this with plants. We're doing this with many types of objects. So most of these data come from a large federally funded program that we have that I lead, actually in collaboration with Ed, but also with colleagues that are at more than 25 institutions around the United States. They are almost every museum you've ever heard of in the United States that has a natural history collection are part of this project. And our goal was to create a 3D representation of every vertebrate genus. So just for reference, we're in the genus Homo. Frogs outside are in the genus Rana. So there's many species in the genus Rana. So our goal was just to get one representative of that genus as a 3D model online. Why just one? Well, we have 10,000 of these genera to scan from around the world. So trying to do every single species is a pretty tall order, but we focused with just one of each genus. We use lots of different things like CT scanning mostly, but also some of these light scanning and photogrammetry now. And this is really, I think of this, it's not really a research project. This is a goal of creating better access to all of these objects that we have across museums in the United States. So today, so far, this has involved more than 200 people across these 25 institutions. And I think one of the things that's, to me, been most satisfying is just for the institutions involved, more than 2,000 students at these universities have interacted with these in class. That might be that they were actually using the real data to create their own 3D models, or it might be that they interacted with a 3D print that was in a lab exercise or something like that. And I'll give some other examples of that. This is just maybe more detail than you need, but just to give you a sense, so we have about 12,000 of these specimens up online now. They're freely available for research, education, art, basically anything that's not commercial, which if you have questions about, I can answer at the end. During the last few years, these have been downloaded well over 100,000 times at this point. But more importantly, you can actually just look at them online. And so because you can look at them easily online without downloading them, most of the impact right now is from people simply looking at them in a web browser like Chrome or Safari. And at this point, we've CT scanned, almost all of the amphibian diversity and the reptile diversity. Maybe that's not a surprise if me and Ed are leading it. But we also have made great progress, especially on mammals. And then fish is like a never ending, there's just a lot of fish, turns out. So one of the interesting things I'm sometimes asked is why we're always so low on scanning birds. You know, there's often a really large community of people interested in birds, especially birders, amateur ornithologists. One of the reasons, as I showed that picture earlier of the Smithsonian with all those drawers pulled out of bird skins, there's nothing inside them. Right, so CT scanning them is going to show us wooden sticks and the stuffing that's used inside those bird skins. So in preparing those skins, actually ornithologists have spent decades taking the things out of the inside of the skins, which is just the feathers, right? So of course, for CT scanning, we want to see the insides of them, but they're just not there in most bird collections. So it turns out it's very rare to find these fluid preserved birds that preserved all that internal anatomy. And so this project was actually quite enlightening for some of our ornithologists that were collaborators, because they suddenly realized that there's a lot of birds out there that we don't have these type of resources for. And it's created some new partnerships between museums and, say, wildlife refuges. There's a famous, I'm trying to see, it's a waterfowl park. I think it's in South Carolina, where they have, this enormous number of the world's ducks and geese on display. The museum here now has a partnership with them. So when animals die, they get put in a freezer and then they end up here at the museum. And because of that, the museum now has some of the very first representatives of the inside anatomy for a lot of those species of ducks and birds. So just to give you a snapshot, these institutions that are participating are spread all across the United States. We have a little bit of an absence here in the sort of Great Plains, but there's also not really major museums with collections in this area. So just move on past this. Just to give you a quick snapshot, so one of the ways our project works is we had an initial project, but the National Science Foundation allows other partner projects to also be funded later. And so we have several of those. We have projects that have focused only on giant things, so things that would never fit in a CT scanner. We had a project that did light imaging of every single bone in a whale skeleton, and lots of whale skeletons and hippos and horses, stuff that we could never fit in a CT scanner. And they also don't fit in jars, so we don't have fluid preserved representatives. We have projects like this one here that spend a lot of time generating images for rodents, and rodents are a really interesting group for mammalogists that study ecology and evolution. We have projects like this that focus only on very, very tiny reef fishes, bird skins, Galapagos tortoises and things from the Galapagos, things from say the middle of Mesoamerica, so throughout Mexico and Central America, bats. All of these partner projects are adding yet more types of data to this sort of large resource that we're creating of these 3D objects. And many of these, as you can tell, are specialized on a particular thing. So just as one example I'll show you, talk about is this, the Galapagos tortoises. So in San Francisco, where I was previously, it has the world's largest collection of Galapagos material. So the largest collection of Galapagos tortoises in one place. The Darwin's finch collection that you probably heard of when Darwin went on the voyage of the Beagle, he spent a lot of time observing and then remarking later in the Origin of Species on Darwin's finches. However, who really made them popularized as a system for studying ecology and evolution wasn't really Darwin. It was someone named David Lack. And David Lack worked on the California Academy of Sciences collections of finches from 1905 and 1906. So this project is actually making all these tortoises, Darwin's finches, lots of other reptiles and things that live on those Galapagos Islands available for researchers, but also available for people that might be interested in learning more about evolution in the classroom. So we put all of these data online, just to give you an idea of what that looks like. So we have something that's basically a repository where we store all of our data online. That's called Morphosource. We also have a program here called Sketchfab, and this is where we actually put all of our sort of colorized models for the classroom, which I'll show you more of in a second. And one of the things that's really nice about our data repository is now you can actually manipulate these X-rays online. You don't have to download them. if you've been at the doctor's office, like I've had knee surgeries, we look on their computer at the MRI or the CT data, and they're showing you maybe where you have an issue in your knee, but you're not walking away with all of those data, right? It's a lot of data. You wouldn't necessarily have the software to analyze it or work with it at home. But now, because we can do a lot of that online, it really makes it more accessible, not only to scientists, but to students that are interested in anatomy. Perhaps not surprisingly, the COVID pandemic turned out to be important for these data. As soon as the world shut down in March 2020, it shut down for all the students in classrooms. It's shut down for graduate students. It's shut down for scientists around the world who suddenly couldn't visit museums. And guess what? There was also no one in the museums to have visitors. There was no one in the museums to send specimens on loan. So for our large community of people that use this resource, suddenly there was nothing that any of these students and scientists could do. It turned out that because we had so much data already online, that there was suddenly an enormous interest in actually using the material that we had put online in our online repository. More than half of all the downloads that have ever come from that repository came after March 2020. So there became this enormous interest in using these data. And this is a good thing. More than half of these data are used for science. That's what the National Science Foundation funded us to do, was to create data for science. But to me, the thing that, as a museum scientist, that's most exciting is to see what a large percentage of these data are used for education and art and other personal uses. Personal uses includes people just love skulls and want to 3D print their own skull of something and have it on their desk. So it's a small percentage of people, but there are people out there that are doing that. So that's exciting. It's exciting because these are people that otherwise would never come through the back door of our museum to look at and use the things that we care for. I'm just going to give you one quick example. So here's a platypus. So if you look online, there's about 100 skeletons of platypi in scientific collections in the United States. So as part of this project, we were talking about Berkeley earlier, we put a specimen of Berkeley's platypus. This specimen, you can't read it. I can almost barely read it. This is from 1922. So this platypus has been sitting in a jar at Berkeley for a very long time at this point, 100 years. We put a CT scan of this online here, and at this point it's been downloaded. That specimen has been downloaded from countries around the world at least 300 times, I think probably approaching 500, little old slide. And the 3D model of a platypus skull that we put up online here, this is also from a while ago, but at this point was viewed 3,500 times, right? From people around the world. And I just want to put those numbers in perspective. So that skull was viewed 3,500 times, and in US collections, there's only 100 skeletons, right? So right here, we've dramatically increased the impacts of, people wanting to understand a platypus. And I'll just tell you, mammalogists at the very least really care about the platypus. So having an easily findable specimen online has really been important for scientists, students, and more. Okay, just a few examples. I can't quite see what time it is with the light off. Ooh, all right, I'll wrap it up. So these are just a few examples of what people tell us that they're using their data for. And I'll read them for you quickly. I'm a master's of research student collecting landmark data for bat skulls. Landmarks means they're putting points on the skull to understand their shape. I'm a paleontologist from China working on a fossil frog. As a frog biologist, this is very satisfying because for paleontologists working on frogs, they've often not had access to all of these skeletons of all the frogs of the world to do their science. And so, now they can do that online. And we also have examples like this where people are understanding more than just what the species is, but they're understanding how it works, right? Because you can use these data as an engineer might use data for a bridge. You can understand how the density and shape of those bones means that they would be resistant to bending or how they would be resistant to breaking. At this point, there's well over 200 scientific papers that have been published using these data. So we know we're making an impact in the research community, which is great. But importantly to me, we're also making an impact elsewhere. So these are examples of connecting these data to students and teachers. So I'm a high school, and an important part here is all of these are in another country. That was why I pulled these examples. You know, I'm a senior high school teacher in Japan. I want to use these data for a lesson. I'm a lecturer in zoology at Manchester Metropolitan University. That's in the UK. I'd like to use these for teaching third-year vertebrate evolution. I'd like to 3D print this model of a platypus skull, that same one, to use in biology lessons at a high school in the Czech Republic. None of these are people that would ever be using our specimens at all, right? And so by putting them online, suddenly that one platypus at Berkeley is, you know, reaching someone in the Czech Republic teaching high school students. And that's, to me, pretty exciting. Just to give you an examples of what this might look like in a classroom. I have colleagues like Chris, who's at John Carroll University in Ohio, having students 3D print weird skulls and try to figure out what all the bones are and writing papers about that for comparative anatomy. I have students in my lab that are artists at heart, so they, even though they're biology students, they're very interested in art. They spent a lot of time making 3D puzzles out of skulls, which is extremely time consuming, but I'll also say is really quite fun because, you know, as a scientist, we can 3D print things and look at them, but as scientists like to take things apart, right? They like to look inside of them, see how they work. So to try to puzzleify a skull was a pretty cool exercise and something that really has played well in the classroom, enabling, you know, undergrads to take these things apart just as a scientist might. We also use, you know, iPads alongside physical specimens in the lab. So here's these are two iPads, and the reason that there are iPads there alongside, you know, turtle shells is that sometimes there's things that we just can't show with a specimen. These are all specimens from the museum's collection. This is part of what a lab looks like on turtles when I teach amphibians and reptiles. We put these iPads alongside because we can actually show digital models that, you know, complement the things that we otherwise are showing them in class. So it can be a way of extending the impact of a teaching collection. We create these types of 3D models. So this is online. This is just me making a video of something that was online. This is a skull of a burrowing amphibian. We can put points on it and put anatomical terms. We can put pictures on it as well. So these are great learning resources for students learning anatomy, students learning about the biology of different animals. We can do crazy things like this. So this is a postdoctoral scientist that works with Ed Stanley and I, creating 3D models here. This is of the tuatara, which is from New Zealand. This is a very rare lizard relative. Usually there are not good anatomical resources for this really sort of rare, critically endangered species from New Zealand. So we can create these animated, easy to use models online where students can learn anatomy from them. This is Ed. This is actually Ed in real time, just to give you a sense of how quickly you could learn to use these real data. These are the real data from a CT scan from a frog. We train high school students to do this, undergrads, grad students. I one time had my 11-year-old trained to do this, to create 3D models. They use those 3D models to print just because they're interested, or to use those 3D models to ask a research question. It seems sophisticated, and it is, but at the same time, it's a toolkit that's learnable quickly, and even from, you know, an early age. So we regularly have high school students working with us, learning this and creating these research data alongside graduate students. We have a lot of software that's used for these, some of which are extremely expensive, but importantly now there's free software as well. And so the combination of free data and free software makes a lot of science happen today. One of the things that Ed and I have been doing over the last few years, it's been very fun, has been to work side by side with teachers. So for the last few years, we have brought in teachers, mostly from the state of Florida, but also from elsewhere. We have brought in middle school and high school teachers to spend time with us to create learning activities for their classroom. And this was important rather than have Ed and I, who I can say are wildly unrealistic about what you can do in a classroom. It was important to have the teachers really inform us about what was going to work for them in their classroom, for the ideas that they wanted to convey to their students in the classroom. So we've created learning activities using these 3D data to bring either 3D prints or digital models into the classroom for these teachers. One thing that we started doing during the pandemic sort of afforded by the fact that we couldn't physically bring people together for this workshop, was we started inviting scientists to participate virtually from elsewhere, basically doing these workshops all via Zoom. So we had these 6 scientists mostly from the US, but also Hillary is at Carleton in Canada, work for a whole week with teachers to develop learning activities about their science, and they study different groups of vertebrates, from fishes to snakes to weird shrews to cassowaries and birds to amphibians and fish, and have teachers basically create learning activities for their classroom kind of focused on that science, but also focused on the scientists. Because one of the things we discovered in working with teachers was that they were as interested in the research and the science as the scientists, to know, how did these scientists get here? Why do you study fish? That's super weird. To be able to explain to their students, how did these people get where they are? And so we've spent a lot of time, creating these relationships between scientists and teachers. And then all of these things now go online. And so we have learning activities. This is actually a learning activity that Ed developed with a teacher who's in Ohio that's on armored lizards. We create these 3D models that are much more focused on teachers and education. This is one to show the sort of evolution of the arm. And, this is fun for me because it's really a relationship with teachers that creates the sort of direction of where we're going with these models. And it gets our specimens, these are all real specimens from museums, gets them into the classroom, which is quite cool. So last, I'll just say the other fun part is that it's created opportunities to reach people that we would just never interact with. And so a lot of these are artists. And so natural history specimens have always been used by artists. If any of you have ever been in art class, possibly you've sketched things like a turtle shell or plants. So those natural history objects have long been used in art. Here's an example of someone working on a scaled display sculpture for the Natural History Museum in Kuwait. I promise you, no one from Kuwait had probably used this specimen ever before, but the digital version gets used. I'm making a series of paintings related to the impact of human activity on bird populations. Or I'm a visual artist working on a research project involving animated shadow projections. We have all sorts of interesting people show up. I'm almost done. I'll give you my favorite example because it involves a frog. So a couple of years ago, I was contacted by Margaret Honda, who is a visual artist who is doing an exhibit for the Carnegie Museum of Art in Pittsburgh. And I can't explain why she was doing this, but this is what she wanted to do. There was a famous painting from the early 1500s by Bramantino that features this frog. Why does it feature a frog? Well, because frogs, in a biblical sense, were viewed as bad. This was actually a symbol, at least in the correspondence from her, this was a symbol of sort of evil and bad things in this picture, right? Just as I talked about in the very beginning. And here is this five-fingered, it doesn't have the right number of fingers, but anyway, five-fingered frog on the floor here. And so she wanted to create a giant 3D replica of this frog in the painting. And here it is on display in the Carnegie Museum of Art. And because she's an artist and she strove for realism, it also has internal bits to it. It has an internal skeleton and anatomy to it that is accurate because it's actually based on a frog in our museum collection, which is wild. I'm not even sure quite what I think about it, but I also think it's quite cool that someone, this frog was on display in the Carnegie Museum of Art and is modeled on a frog that's somehow in the collection at the Florida Museum of Natural History, and just made it possible because we've been putting these data online for really anyone to use. It's a great example of that. All right. And last, I'll just say this, we've been doing this a lot for the past 7 years at the Florida Museum. We have an enormous amount of these data that we make available. If you've been through the Florida Museum of Natural History recently, and if you haven't, you should, there is a really nice temporary exhibit about paleontology at the museum. It has a living lab where there are people actually doing science. They're preparing fossils on exhibit. It's pretty fun. Off to the corner, you will see a giant screen where you can manipulate 3D objects on it. Well, that all comes from our work. Those 3D objects are versions of real specimens in the museum's collection that we've put there, not only because you can manipulate on a screen in the exhibit, but we can also do that online as well. And so we're reaching people well beyond the walls of the museum now. It's great. And I'll leave it there. If people have questions, happy to. Otherwise, you're free to send me an email if you have other questions. And thanks very much for having me here today. Got it. Okay, great.
That image of a frog with all the bubbles in it, are those eggs?
Yep, those were eggs. Yeah, one of the neat things is that we can see eggs inside these animals. Just as a quick example, I have a student that was working on marsupial frogs. Marsupial frogs carry, the moms carry the eggs in a pouch on their back. But because they're inside mom, even though they're developing, so they get laid, they get fertilized, and they get put back in this pouch, we can't see them because they're covered with skin. And so one of the really exciting parts about being able to see them in X-ray was we can actually see the babies developing through the mom's skin, which we'd otherwise never be able to see. And for some of these species that are already extinct, right, there's never a chance for us to get them in the lab to understand their development. The fact that we can see through their skin in an X-ray in a preserved museum specimen is pretty exciting. Yeah.
So you could probably see this chat
Yeah,
question.
let me read it real quick so I know. I'll read it for everybody in case you can't see it. As amphibians and reptiles are seriously endangered by loss of habitat and global warming, hundreds of frog species have been lost since the 1970s, and the prognosis is not good for many more. Aside from accruing and sharing important scientific knowledge about the biology of frogs and amphibians, does the museum actively have a goal of helping the viability of these species in the wild? Yeah, sure. One of the reasons I became interested in studying amphibians was, especially as someone who was a paleontologist who only studied extinct things, was this sort of seeming contradiction between discovering new species and extinctions in the wild happening at the same time. And we are in the point now where we are actually describing species as new to science that we think are already extinct in the wild, right? Because sometimes these things sit on the shelves of museums for 50 years. In that intervening time, they've gone extinct until a scientist who knows enough about those animals looks at them and says, whoa, hey, look at this. This is a new species. This has never been described before. And those species are already gone. So it really is an acute issue for us. So we do projects in my lab that do focus on conservation and understanding their biology. So many of these species, just as first principles, we don't know anything about their natural history. There are frogs that breed in all sorts of types of water. There's frogs that don't have tadpoles. There's frogs that have different types of microbes that live on their skin. Understanding all of those basic aspects of biology is super important if we're going to actually come up with plans for conserving them. Just as a very obvious example, you know, if frogs depend on different types of water bodies for their survival because they lay, you know, tadpoles in streams, tadpoles in ponds, tadpoles in bromeliads, tadpoles in tree holes, tadpoles, all sorts of different places. If we don't understand that critical aspect of their biology, how are we going to make a plan for, you know, how we're going to actually, what piece of the habitat we're going to protect? So we do a lot of work also with organizations that make conservation plans for species in the wild. And a lot of the biology we do both in the field and the lab helps to inform those plans going forward for conservation. Yeah.
Have you used artificial intelligence to go back and look at the skeletons that you have to see if what someone thought was a new species was actually the same that you had in a different lab somewhere?
Yeah, that is a great question. We are standing right on the edge of that right now. A lot of artificial intelligence for dealing with images has focused on two-dimensional images. We have a lot of three-dimensional data. And just now there are some developments happening mostly in the biomedical fields of using three-dimensional data, like for instance, MRI data or CT data that are taken from patients. Just now we're on seeing the development of AI tools for those type of data. And it is something that we are very excited to take advantage of. So we haven't done it yet, but I'll also just make one point. Until we had so much data, there was also no reason to use an AI tool, right? Because we could look at 20 things pretty easily in an afternoon, but it's a real pain to look at 10,000 things in an afternoon. And so, yes, one of the things we're doing right now is sort of making those collaborations to see, how AI tools could help us sort through issues like you mentioned right now about identifying, misidentified species in the past. Sure.
This question has to do with education.
Sure.
Since that's a large portion of the pie there. I'm curious about the language of scientists.
Uh-huh.
Is there a universal language, for instance, to the Chinese high school science teacher wants to get information, downloading, and or just communicating with you here and you going to Africa? I understand that all the phylums and the species get names which are common, but just communicating in general, the teaching techniques and explanation of the data, there's a lot of language to be used there. How can you communicate effectively?
That's a great question. So has a lot of answers to it, in fact. So this is something that I think we're talking about a lot in the museum community right now. To be straightforward, the sort of language of science, for better or worse, is English. The vast majority of science today is published in English, regardless of where you are in the world. We're fortunate because I learned English from a young age, so it's easy for me to publish in English. But that is not true for many of the colleagues and collaborators that we work with. So regularly in a year, I might publish with people in 15 to 20 countries. That's just a normal year for our lab. So trying to collaborate and communicate to one another where everybody has different home languages is challenging sometimes. But when it comes to educators, this is a big challenge for us. And so one thing that we've been doing recently from the museum websites is seeing, what are the countries that most often are using our data and what languages do they speak? So I can tell you right now, we have a move afoot to begin translating some of our resources online into Spanish, which makes good sense being the United States. But Portuguese and Chinese are probably quick behind that because we, especially in Florida for a lot of our museum collections, we have a lot of Portuguese speakers, mostly from Brazil, using our data. China is another one. The one nice thing about Chinese academics is they tend to speak English quite well and write quite well. But that doesn't actually, it's actually interestingly not true for a lot of South American countries where, for instance, I have a Chilean student who arrived 2 weeks ago. His English is not great. So we have Spanish speakers in the lab to try to kind of facilitate communication a bit better. But this is a real challenge, I think, as you just put your finger on, of trying to figure out the best ways to communicate. There has been a recent push, actually, suggestion to start using AI to help better translate scientific resources across many languages quickly. And maybe that will help make some of these scientific studies more accessible. For me, a lot of the literature I work on just as a professional is in French. I mostly work in Central Africa, so a lot of the historical literature is in French. But we regularly are dealing with German, English, Portuguese, Spanish speakers, Chinese speakers as well.
Yes. Hello. I have a question that it's been wonderful to see. Is this better? Okay. It's wonderful that the students today have access to all this that we didn't have back then. But my question is on invertebrates and frogs and so forth, what is their pain threshold? When you're working on a lot of them when they're alive. Do they have a pain threshold? Yep.
So actually, it's a really interesting question. It used to be thought that for especially amphibians and reptiles, that it's phrased in different ways, but perhaps they could not feel pain or appreciate pain in the same way that we can. There's definitely, I'd say, compelling evidence that that's not the case. that they can feel pain. You know, so when we manipulate these animals, especially when they're coming back here as a specimen, you know, these are euthanized. They're euthanized in a humane way that basically makes these animals go to sleep. But for other experiments, we have ways of trying to reduce their pain. So just as a quick example, I have a student who spent part of her dissertation doing some surgeries on frogs. And it was simply to do some little implantations of little metal beads so that she could see them move in x-rays. And to do that is complicated. It involves a lot of attempt to try to cause these animals as little discomfort and as little pain as possible, which you might imagine for a frog is hard to gauge. You know, is it an uncomfortable frog? All toads look uncomfortable no matter what, right? They all look uncomfortable. So, you know, we spend a lot of time, you know, looking at the biological signs of discomfort and pain and trying as much as possible to limit those. And yeah, it's a good question. Thank you.
Thank you. There are 2 more in chat.
Oh,
Dr.
I'm sorry.
Blackburn?
Sure. Do you have 3D data for the Chilean toad, Calyptocephalus? Wow, this is specific. Do you have 3D data for the Chilean toad, Calyptocephaloa gayi? I've used this study to study the effect of peptides hormone on blood pressure and kidney function. Yeah, we do. We do have data for Calyptocephaloa. I was in Chile in February and I got to hold one in the field that we caught, which was an amazing experience. Yes, we do. Send me an email. I will be happy to connect you with some 3D data if you'd like to look at one. In fact, we have this Chilean student here right now related kind of to the Chilean toads, but also to my trip there in February. All right, the other, moving here to live next to a pond in Gainesville, I was amazed to discover 4 different types of frogs. They have managed to arrive at this little pond. I cannot explain, but it's a wonder. Green tree frogs, southern chorus frogs, American bullfrogs, and pig frogs. Yes, they will seek out any water. We have a pool in my backyard, which has chlorine in it. I think we've caught 6 species of frogs in my backyard, which is amazing. You know, one of them I've actually only seen on 2 occasions, which are the spadefoot frogs. They only come out after really torrential rains. So it was there. Some of you might know who Archie Carr is, who was a famous biologist at University of Florida. Archie Carr has this great line that spadefoot toads depend on erotic thunderclaps. Only after an erotic thunderclap will you see them. So I'll bet there's actually more than 4 frogs in your backyard. Just some of them are only come out occasionally, so they're hard to see. All right. Yeah. And I'm happy to stick around. I'm having lunch here as well. So if you have other questions, feel free to let me know.
Okay.
Oh, one more.
Going back to language, is the Latin still, can that be used by the Chinese? Because they know they use Latin for naming of the different species.
Yeah, so one currency in biology that we all do have in common are the scientific names. So scientific names, at least historically, were in Latin or Greek. So we do have that in common, although, you know, there has been a move to use other languages even in scientific names as well. A lot of anatomical terms, you know, Latin is very important for an anatomical term. So I would say if you know Latin, you've made some progress, but of course, you know, very few students are trained in Latin now, so it might not actually be quite as helpful. In my lab, we actually spent a lot of time because many of the new species that we describe are from sub-Saharan Africa. We actually spent a lot of time using local language names for the species, and part of that relates to conservation. So I've done things where I've had groups of village elders come up with a scientific name for a species, in part because, well, that species literally only occurs there. Having a little bit of community buy-in where they actually helped create the scientific name for that species, you know, moves us a little bit further along in terms of having them have an investment, you know, in that species that only occurs in that one place.
Thank you. John Axe, final thoughts?
Yes. Am I heard?
Yes.
Heard?
Yep.
Okay. Well, this completes our 6-talk series from the Museum of Natural History. And I have to admit that 6 weeks ago, when I thought of the Museum of Natural History, I thought of Hull Road and all these wonderful displays that they have that we've often gone to, the Butterfly Garden being one example. But what I've learned, and I hope that people that have attended these lectures have learned, is that Hull Road is just the tip of a very large iceberg. And the people at the university museum are so enthusiastic of what they do and so anxious to tell us about it. So I want to thank Dr. Blackburn again for the talk. But this doesn't complete our planned series, because the first speaker who I contacted was Dr. John Brock to speak about the age of the dinosaur and its abrupt end. And he had to cancel, unfortunately, because of a death in his family. But in our fall season, which begins the first week of October, is going to include a session on paleontology organized by Phyllis Saarinen. And we're hoping to re-invite Dr. Block for this series. So we hope to see you in October. And let's thank again Dr. Blackburn.
Thank you very much.
Best series.
That's very nice.
Who's that?
Oh, thank you.
Holly Farish Hunt.
Once about
Sorry?
Holly Farish Hunt said that.
Holly Parrish.
over
She's been
some
involved for
period of
years.
years. People observed